Welcome to Chapter 15. This chapter is the first chapter in our last unit. You've almost made it! In Chapter 15, we will be learning about intracellular compartments and transport between these compartments. Before we get too far, I want to point out that Unit 4 contains a hodgepodge of materials, so it is important that you understand the objective of each chapter before we move on, because we will not be directly building upon the information in each chapter. As you can see on this slide, here are our chapter objectives. These are the objectives you will need to have mastered by the exam. Please let me know if you need any help in meeting these objectives. Here is the outline for this part of the chapter. We are going to start by identifying the membrane-enclosed organelles of interest in this chapter. Here is the outline for this chapter. We are going to start by identifying the membrane-enclosed organelles of interest in this chapter. We will then talk about protein sorting. As you should be aware of by now, proteins perform a majority of the functions of the cell. It would then follow that most of the cell's actions will be to move proteins around to get them to their specific locations. We will then wrap up this lecture by reviewing vesicular transport with two examples, the secretory pathway and the endocytic pathway. Now let's get started. As you have started to recognize through this course, the amount of activity going on in the cell at any given time is mind-boggling. Sometimes these processes work at crossed purposes, meaning that while the cell may be performing a catabolic reaction in one area, it may be performing an anabolic reaction somewhere else. This is where the organelles come into play. It could get quite messy trying to do these reactions right next to each other. The eukaryotic cell has developed a solution for this by evolving to contain several membrane-enclosed organelles. So far in this course, we've only really talked about three organelles: the nucleus, the mitochondria, and the chloroplast. But, as talked about in the first chapter, the cell contains many more organelles. In this chapter, we are going to explore those organelles and how they function. Let's start our conversation by first getting acquainted with the organelles of interest, and then we will discuss the evolution of these organelles beyond the endosymbiont theory. On this slide, you can see the list of the main functions of the membrane-enclosed organelles we are going to focus on in this chapter. This slide is very important. Make sure you study this chart thoroughly. Let's talk about each of these compartments before we start looking at pathways. The first of these compartments is the cytosol, which is bound by the cell membrane. As you know, the cytosol is the jelly-like substance that fills the main cavity of the cell. The cytoskeleton and all of the organelles are suspended in this jelly. As you know from Chapter 13, glycolysis occurs in the cytosol, but there's a lot more processes that are also occurring in the cytosol. Also, many signals being passed from the outside of the cell to specific sites inside the cell are passed through the cytosol. The nucleus is the dominant membrane-bound organelle. It is surrounded by the double nuclear membrane, and it contains nuclear pores that allow for molecules to pass between the nucleus and the rest of the cell. As you know, the primary role of the nucleus is to contain the DNA. Connected to the nuclear membrane is the endoplasmic reticulum, or ER for short. This is a continuous membrane, so it is really impossible to separate the two or identify where one starts and one ends. The ER is a series of connected tubes and sacs that can be found throughout most of the cell. As you know, there are two sections of the ER, the rough and the smooth. The rough ER, or RER, is where some proteins are synthesized. These proteins are generally destined to be housed in other membranes and not operate independently in the cell. The smooth ER, or SER, is responsible for the synthesis of hormones or other lipids, as well as the catabolism of toxins such as drugs or alcohol. The SER is responsible for the production of all new membranes within the cell. We will be revisiting the ER in Chapter 16 when we talk about cell communication. It plays a significant role in the communication. The next organelle that we are going to talk about is the Golgi body, which is our shipping center in the cell. It can receive, sort, modify, and package proteins and other molecules to either be exported to the cell membrane or to other organelles. This is where carbohydrate modifications are made to proteins and lipids destined for membranes. It is important to note that the Golgi body has sidedness. One side of the Golgi will be receiving vesicles, absorbing the vesicle, and releasing its contents into the lumen of the Golgi. When ready to be shipped out, the other side of the Golgi will bud off and depart for its destination. Let's look at our last three organelles as a comparison. These are the somes: lysosome, endosomes, and peroxisomes. It's easy to get these three mixed up, so let's make sure we get them straight. All three are relatively small sacs, but each performs a very important function. Lysosomes are responsible for the degradation of cellular components. For example, if a mitochondria becomes leaky for whatever reason, the lysosome is responsible for degrading it into building blocks that can be used again. In contrast, peroxisomes are responsible for the breakdown of lipids or neutralizing toxins. And then what do endosomes do? They are responsible for sorting the materials that are brought into the cell by endocytosis. We will talk about what the process of endocytosis is here in a bit. I know we didn't talk about the chloroplast and mitochondria, but after chapters 13 and 14, I figure we have these pretty well understood. Please refer to these chapters if you need a refresher on these organelles. Before we move on from the organelles, take a moment to appreciate the components of the cell. As you can see from this chart on the slide, each of the organelles we talked about take up a specific amount of volume within a typical cell. It is not important information for you to memorize, but do appreciate the different volumes that each of these consume within the cell. We've already talked about the endosymbiont theory in Chapter 1. This is the theory that a prehistoric eukaryotic cell phagocytized the mitochondria and chloroplasts, both of which were prokaryotic cells in their own right prior to being engulfed by the eukaryotic cell. Remember that there are two pieces of key evidence to support this theory. The first is the presence of a double membrane, and the second is the unique DNA contained within each of these organelles. If you would like an in-depth refresher on the endosymbiont theory, please refer to your textbook. On to the endomembrane theory. Not all organelles have double membranes, which means they could not have arisen in the cell via the same process. These organelles are believed to have arisen from a process described by the endomembrane theory. In this theory, it is proposed that the nuclear membrane, as well as all the other organelles we talked about, were formed from an invagination of the plasma membrane. The proposed model is that in the ancient prokaryotic cell, the DNA was isolated by a section of the plasma membrane to keep it separate from membrane-bound ribosomes. This process continued to grow to form the endoplasmic reticulum, the Golgi body, and the other organelles we just talked about. I know this seems a little far-fetched, but we've actually observed this process occurring in modern-day photosynthetic bacteria. Make sure you understand how both these theories work and be prepared to argue the evidence that supports these theories.